Good morning. It's 11 o'clock on Wednesday, October 5th, 2022, and I am Jana Hexter from the Northeastern IPN Center, and I'm delighted to invite you here for our toolbox presentation, Utilization of Biologicals and Biofumigation for Effective Management of Soil-Borne Diseases in Fruits and Vegetables. And I welcome you here today. It's a beautiful full day up here. It's the first of the leaves are turning, and it's turning very beautiful. So I'm delighted to welcome our presenter, Mahfouz Rahman. But before I do that, there's a couple of housekeeping things. So if you could move the slide for me, please. There we go. There will be a recording of this webinar, and anyone who's registered for the webinar will get a copy of the recording. And we do edit the beginning and the end a little bit. So sometime next week, you should get an email from me in your inbox with the recording. And if you don't, let me know. But yeah, that should be coming to you. Next slide, please. We welcome your questions. And just to make it easier for us to keep track of the questions, what I ask you to do is to use the Q&A feature. If you're not familiar with that, if you scroll over your mouse over the screen, you'll see a black box that'll appear at the top or the bottom, a strip. And somewhere in the middle of that strip, there's something called Q&A with two little rectangles. And if you click on there, you can ask a question, and you can do so anonymously. So feel free to check the anonymous feature if you would like to. This way, it helps us keep track of the questions. So if there are duplicate ones, we can mark them as both answered. And next slide, please. Great. Oh, it's my pleasure to introduce Dr. Mahfouz Rahman. He conducts applied research and provides leadership for statewide educational informational programs in plant pathology that include disease identification and management recommendations. Early diagnosis of pest problems remains the cornerstone of his adaptive and applied research program at West Virginia University that seeks to address disease problems primarily in vegetables and fruits. He takes an integrated approach that includes cutting-edge research on pathogen detection and quantification, proactive measures to avoid the introduction of harmful pathogens with planting material, and adoption of measures based on the disease forecast model. So welcome, Dr. Rahman. Thank you for taking the time to present with us today. So the next is we have a couple of questions before we get going. A poll should pop up on your screen. And there we go, here it comes. And there are no right and wrong answers here. It just gives us a sense of who is on the call and your level of knowledge in the call. And so I'll just be quiet for a couple of minutes, and so that you have a time to answer those questions. Great, we've had 90% of you complete the poll. And I'll just be quiet for 1 minute in case, or for a little bit in case the last person is completing, and then I will share the poll with you. All right, so we share the results. So the question, how knowledgeable are you about soil-borne diseases? We have a pretty good split of people who are not at all knowledgeable or moderately. How knowledgeable are you about the management of soil-borne diseases? And again, we have a good spread there across not at all, somewhat, and moderately knowledgeable. Are you familiar with anaerobic soil disinfestation? Sorry for mangling that up. And 70% of you that are not. And how knowledgeable are you about biofumigation, ASD, for soil-borne disease? And the vast majority of people not at all knowledgeable. So, Mahfouz, you have got an audience who is eager to learn, clearly. And so with that, I will turn it over to you.
Thanks, Jana, for the introduction and invitation to share some of my results from previous years' experiments and applied research, more specifically on soil-borne diseases. So I'll give you a little bit of introduction of plant diseases and the different types of diseases. Then I'll show you some of the results I obtained from biologicals and biofumigation, more specifically on anaerobic soil disinfestation. So some of you might know that there are two different types of plant diseases that can affect yield and quality of plants. And in many cases, some of those diseases can be fatal. They can actually kill the plants. So one type is called foliar diseases. So these are the disease-causing organisms that in fact leaves, stems, and in some cases fruits. So they can kill the tissues, and in severe cases under congenial environment for diseases, they can actually kill the plants. But in general, these diseases are relatively easy to control if the detection or diagnosis is done early. And in the management program, if you include efficacious fungicide or bactericide products. Other group of diseases called soil-borne diseases. These disease-causing organisms basically live in the soil. So these organisms are hidden underneath. And in many cases, growers as well as researchers do not give much attention to what is happening with the plants. They are basically seeing plants are drying up gradually and eventually die. There are fungal pathogens, bacterial pathogens, nematodes, and some of the viruses can also be soil-borne. For fungal pathogens, Pythium, Rhizoctonia, Fusarium, Sclerotinia, Verticillium, and Phytophthora, and there are many different species belong to those are soil-borne. Some bacteria, notably Ralstonia solanacearum, that cause bacterial wilt of tomato, and it's a very, very important disease. And when I see growers are losing their tomato plants, those with the green tomatoes, and the plant collapses like in overnight or in some cases in a few days. So that is very, very important disease. And in many cases, we really cannot recommend any disease management option when we see the disease. So we need to be prepared. We need to understand what can happen to our plants due to soil-borne diseases. As you can see, I mentioned that in many cases, soil-borne diseases can cause 50 to 75% yield loss in many crops. And many of the nematodes, obviously, most of the nematodes are soil-borne, except a few. So those also have the potential to cause problem on plants. And most importantly, these organisms can live in the soil, in plant residues and organic matters for many, many years. In case of foliar diseases, if you have one crop and the crop you harvest and it's gone. But for soil-borne diseases, these organisms can be there for many years. In addition, they can form special survival structures. In many cases, those are called sclerotia. These are basically, like clump of hyphae, hard structure. They are resistant to adverse environmental condition and microbial degradation. That's why they can survive in the soil for many, many years. Well, it is difficult to control, as I mentioned earlier, compared to foliar diseases. Even with conventional strategies, here I want to mention that you apply fungicides or bactericides. In this case, for soil-borne diseases, if you apply fungicides in the soil, the success rate can be very, very low because they are protected in the organic matter and plant debris. Only option growers had to use fumigants. So those are mostly synthetic fumigants. Those have been very, 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 very effective, like methyl bromide. And after many of you know that methyl bromide have been phased out. And after phasing out of methyl bromide, growers have tried and researchers have tried many different synthetic fumigants like Talon C35, Picklor 60, Besamid, Bayfam, but not any of those products are that effective. And I'll also tell you why also these products are very difficult to use by small growers. And obviously there is no question that organic growers can use this. So I came up with the response from many of our growers for organic options for soil-borne disease control. And I tested these biologicals and biofumigation in many different commodities like strawberries, especially for black root rot complex and crown rot, for tomatoes, verticillium wilt, fusarium wilt. And in many cases, I cannot rule out that soil had bacterial wilt causing organism there too. But for eggplant and okra, these are minor crop, but I specifically tested these for verticillium wilt and for okra also verticillium wilt. I'll share the results with you. So for strawberries, as I mentioned, the black root rot complex, what you can see at the top left, this is a normal strawberry plant with plenty of feeder roots in it. But at the right-hand side here, you can see these plants are affected with black root rot complex or black root rot disease. And you can see the feeder roots are basically gone, and the root took a red tail appearance with brown coloration. So obviously, if you do not have the feeder roots in plants, those plants will not be able to take nutrients, and plants will be stunted and eventually may die. So why we call it black root rot complex? Because there are many different types of organisms involved with it. Rhizoctonia, Pythium, in some cases, Fusarium, and also nematodes have been found to cause injury to the root system, and then those organisms go and infect those plants. In addition, as I mentioned, the crown rot can be caused by two different organisms. Anthracnose crown rot, this is caused by Coleotrichum gliosporidae species complex and Phytophthora crown rot. So in this case, you will see the total collapse of plants. So if you want to do a quick diagnosis, you will have to uproot the root system and crown and then take a longitudinal cut. You will see the discoloration of the crown. In this case, anthracnose crown rot, you will see brown discoloration with marbly appearance. And for Phytophthora crown rot, you will see dull brown color. But there are other diagnostic methods, especially diagnostic labs will do for you by isolating the organisms from these affected crowns. So here black root rot complex and crown rots, these are in most cases all the soil-borne organisms. If you have planted the same crop in the same soil over many years, these soil-borne diseased organisms will multiply and cause problem. And they can eventually cause bigger loss in a perennial system. We still have perennial strawberry production system in the Northeast. In many cases, crop rotation, we recommend if you have the soil with problems, if you have soil infested with soil-borne pathogens, you rotate. But in many cases, crop rotation is not feasible because many small growers, they have very limited amount of land to grow crops. And you may be familiar that UP growers, they want to grow their strawberries in an area where people can get access very easily, and that is visible from, in many cases, from the roads and highways. But for fumigation, especially for synthetic fumigation, there are many, many problems with synthetic fumigants. there are regulations. Those synthetic fume application of those synthetic fumigants need very large equipments, and small growers cannot afford to get those. And also new regulations prohibit growers to use those synthetic fumigants close to school, church, and any houses in any dwelling. So this is very, very limited to very large growers. And also synthetic fumigants can pose risks to environment and human health. And obviously organic growers cannot use those. And here you can see, so there is a huge critical need for us to find alternative options like biologicals and biofumigations. Here you can see a matted row strawberry production in our Mineral County in West Virginia. What these growers basically do, they plant their strawberries 1 year and they try to harvest strawberries up to 3 years. And in some cases, here you can see this photo actually not showing the dead plant, but even in this case, when I took the photo, actually many areas showed lots of plants totally dead at that time due to soil-borne diseases. So when we are talking about biological control, we need to think about what is the mechanism of biological control. These are basically beneficial microbes we are using for biological control. The basic mechanism is called indirect antagonism or competition. So what happens when you have beneficial microbes, they compete for the sites on the root system. Here you can see these beneficial microbe, Trichoderma, what is shown in green color, is actually taking the root systems, taking over the root system. So harmful microbes will not get space to infect this root. And nutrients consumed by beneficial microbes. So whatever the nutrients coming from the root exudates, they are being taken by beneficial microbes, so depriving the harmful microbes. And also there are other mechanisms of beneficial bacteria or beneficial fungi antagonism. They can attack the harmful microbe and then decay those harmful microbes, hyphae, especially in case of fungi. Antibiotics, they can produce antibiotics and lytic enzymes that can actually kill the harmful microbes. And they can also produce cyclic lipopeptides. Those are in the antibiotics group. And they also can produce chitinases and glucanases. These are the, so chitin and glucan is in the fungal hyphae. So if they produce chitinases and glucanases, they can actually degrade the fungal hyphae. So these are the basic mechanisms of biological control. So what we thought about this biological control is basically from the mechanism that we need to actually put this biological control agent on the root system of the plant ahead of harmful microbes. So in our strawberry work, we tried to pre-colonize the plant root system by growing tips to flag plants in treated or inoculated planting medium. So we had 2 different types of treatments. We inoculated planting mix with beneficial microbes, and we also treated the fruiting field. Here you can see this work was done with 6 different treatments. I want to explain it because it's a little bit complicated, because we did the treatment during the flag plant production and also in the fruiting field. So non-treated, that means we did not treat the planting mix and we did not treat the fruiting field. So that's basically no treatment there. Mustard cover crop, obviously, that is in the fruiting field, we grew the mustard cover crop and then mixed in the soil. Regular planting mix inoculation with probiotic bacteria. Regular planting mix, that means we did not do any changes in the planting mix. Pasteurized planting mix inoculation, that means we pasteurize the medium to kill the resident microbes. So in the absence of resident microbes, can colonize the root system more efficiently. And first rice planting mix inoculation, that was not very effective. So it's a good news for the growers that you will not have to pasteurize your medium. Anaerobic soil disease infestation, I'll talk about this, and synergistic, basically combination of planting mix inoculation and the soil and the fruiting field in disinfestation of soil. So I'll not talk too much about the product we used because there are many similar type of products you can use. So in our case, we used a commercially available product called TerraGrow. As you can see, there are 5 different types of beneficial microbes, especially Bacillus type of bacteria is included in that product. So once again, you can see out of 6 different treatments, number 2 and 5 were done in the fruiting field, 3 and 4 were done in the flag plant production system, and number 6 are a combination of planting mix, inoculation, and fruiting field. So I think many of you have done this mustard cover crop and tried in many different crops. Basically, you grow the mustard in up to the flowering, and 75% flowering. In this case, we used Caliente mustard, and then like mixed it in the, chopped it, and then mixed it in the soil, covered it with the plastic. So, Dana, I think you may have some question at this time.
Yep, I actually don't see any right now, but for those people who've just come in, if you have any questions, if you can put them in the Q&A box, which is in the bottom or top of your screen, and I'll just wait for a moment, see if any pop up. And if not, we will carry on. There'll be another opportunity for questions. So, let's move forward, and then we'll pick up questions towards the end as well. Okay.
All right. So anaerobic soil disease infestation, from the pre-webinar question, I realized that many of you may have not heard about this. This is a relatively new method for soil-borne disease management. So it has been used for many different crops, and especially in California and also in Florida. Especially synthetic fumigation have been used historically for, high value crop like strawberries and tomatoes. So these alternative options also have been used for strawberries and tomatoes and few other crops. So also it's a new terminology to many of you, but I should tell you that this is not really very difficult to do because it's only 3 different steps. At the first step, you have to incorporate organic material or carbon source in the soil. You can use many different types of carbon source. Basically, you prepare the soil, prepare the field plot, and then mix the organic material. In this case, we used mustard meal at the rate of 5 tons per hectare. So to some of you, you might think, oh, this is too high of an organic material amount, especially mustard meal. Still a little bit expensive, but it also adds biomass in the soil. So when you apply this in the soil, you have to mix. In the small plot, we used walk behind rototill to completely mix in the soil, and then covered this area with plastic tarp. So at the same time, many of you, when you are using beds, you are preparing beds, and especially plastic mulched bed, you also put the drip irrigation tape at that time. So in the third step, you basically cover those beds with plastic mulch, and then you turn on that drip irrigation. So you have to, how long you have to irrigate those beds? Up to saturation. So the whole idea here, when you saturate these beds, this air going out of the soil has capacity to hold the air. So when you take the air out, it makes an anaerobic condition of the soil. And then you keep it for at least 3 weeks, 21 days, and some people even tried up to 1 month. So during this 1 month period, these anaerobic microbes, especially bacteria, multiply. They profusely, that population goes up, and then they can kill the harmful microbes in there. And also it can accumulate toxic gas. Those can be suppressive, or a volatile organic compound. Those can be suppressive to pathogenic microbes. So this is basically the way we do the anaerobic soil disinfestation. So in the next few slides, I will show you what has been done in large areas in California. So as you can see here, they used rice bran as a carbon source, and we used mustard meal, but some other growers tried grape pomace as a carbon source. So basically they sprayed it in the top of the soil and then incorporated it. And this is showing the second and third step, covering the beds with plastic tarp and then irrigate it through drip irrigation system. So when you are doing the irrigation, you have to make sure also these beds remain saturated for up to 21 days. So if you have used synthetic fumigants, obviously you know that after a certain period of time, in most cases 10 days, you have to go and then cut the plastic to remove any toxic gas in there. Because if you have toxic gas and then you are planting your seedlings, that can actually kill those seedlings. So the same thing for precaution, we do the same thing 24 hours prior to planting. We just cut the plastic, the holes, and then let the toxic gas going out. So this is the result I'm trying to share with you from California trial. Here you can see they did the anaerobic soil disinfestation with 9 tons per acre of rice bran. That is very high, but rice bran is relatively less expensive. That's why they could use that kind of amount of rice bran. But here you can see the result comparing the untreated one and anaerobic soil disinfestation. In the untreated one, verticillium will almost kill many of the plants, or in the verge of killing. So now I'm going to share some of our results, what we have done with strawberry trials. The first thing we have noticed in our treated plots in the spring is the number of weeds coming through the planting holes. So as we use the plastic covers, weeds did not grow in the whole plot, but through the planting hole, we saw many weeds coming up, and then we enumerated those weeds. So here you can see for plot basis, in 2 treatments, basically that received anaerobic soil disinfestation, significantly lower number of weeds in those plots. In mustard cover crop, we saw numerically lower number of weeds, but it was not significantly different with non-treated control. So many other workers and researchers also reported very similar result, that anaerobic soil disinfestation during that process, the weed seeds can be killed. That's why we can see less number of weeds in ASD plots. So the first year fruit harvest, as you can see, we got up to 600 grams per plant of strawberries, which is very typical in organic system in the Mid-Atlantic and in the Northeast. So here you can see in the first year, the difference was not very, very significant, but we saw significantly higher only in ASD plots compared to other treatments and non-treated control. But the real difference we saw in the second year, because many of our plants actually died in the first year and the second year. Here you can see we started with 20 plants in each plot. Here you can see the non-treated line is blue line. And at the end of the first year, actually, we saw many stunted plants and also up to 6 plants died after the first year. And after the second year, also many other plants. So we almost lost 50% of the plants in non-treated, followed by mustard cover crop. But the lowest number of plant mortality we noticed in TerraGrow treatment and ASD. So here you can see the results from the second year harvest. We have found like significantly higher strawberry fruit from all the treatments except mustard cover crop and non-treated control. So, but overall, you can see the amount of strawberry we harvested in the second year was very low. It's not only some plants died, also during this because of the spring frost. So many of our flowers actually died due to the spring frost. So, we can definitely see here that anaerobic soil disease infestation and combination of those very, very promising here. So at the end of the second year, just prior to the termination of that trial, we took photos of those flowers, as you can see here, non-treated flowers and Caliente mustard cover crop. And here in the middle, the TerraGrow treatment, regular and pasteurized. And here ASD and combination of ASD and biologicals. Here you can see from the photos that combination treatment really had very good plants with high vigor at the end of the second year. So we also did some study on by collecting the rhizosphere soil and analyzing the nutrients from that. As you can see in this from this table that in the mustard cover crop, obviously phosphorus and potassium content was higher, significantly higher, because we added significant amount of biomass there. But interestingly, here you can see when you use the TerraGrow, potassium content was higher. Because these beneficial microbes, these are also called probiotics, so I'm not surprised because they have the capacity to solubilize the nutrients from the soil and make it available to the plants. But once again, to ASD alone and ASD combination of ASD, we can see the significantly higher amount of phosphorus and potassium. Magnesium was not significantly different in different treatments. And soil pH it is not very easy to change the soil pH in 1 year or 2 years by applying this kind of organic materials. So, but organic matter content in ASD combination treatment was significantly higher compared to other treatments. So we also tried to understand what has been happening for the plant stunting and plant mortality. We found that low vigor due to black root rot complex. And we also found some plants died due to Phytophthora crown rot. We did not get any Coleotroticum crown rot. Nematode population, we did not see any difference, but we are trying to enumerate the beneficial microbes in the rhizosphere, which will take more time for us to complete. So to summarize these results from strawberry study, we might say that finding suitable alternative of synthetic fumigants may be difficult. It will not be the real alternative of synthetic fumigants. Still, synthetic fumigants can be more effective than these biologicals and biofumigation in general. But probiotic bacteria may provide benefit to strawberry plants if we can let the strawberry root system colonize ahead of time before taking it to the fruiting field. But the most important take-home message from this study, I will say, synergistic effect that we found from the probiotic bacterial inoculation of flag plants and then followed by anaerobic soil disinfestation of the fruiting field. So what we'll have to do, we are trying to unravel some of the mechanisms involved with this synergistic effect, and that will take more study and then more work in the future. I think we still have.
Yes, we have lots of questions. All right. So the first one, it's kind of implied, but it's a good question. So do the beneficial soil microbes survive the anaerobic process?
It's about soil beneficial microbes.
Do the beneficial soil microbes survive the anaerobic process?
That's really a good question. So this is, I don't know exactly what can happen to these beneficial microbes, because we did not take these beneficial microbes during the anaerobic soil disinfestation process. But during this anaerobic soil disinfestation, what we do, we let the anaerobic microbes to multiply. And also during that process, there are some, volatile organic compounds and other organic compounds can be released from during that process. The whole mechanism for this beneficial effect we are getting is not known at this time, and that's why we are trying to understand more. But my take on this will be this process can also kill some of the beneficial microbes. But in most cases, if you can kill the harmful microbes and you still have the beneficial microbes, this is basically the ratio. If you have more beneficial microbes in the soil than harmful microbes, then basically beneficial microbes will take over and then suppress the harmful microbes. So it really doesn't matter either. Some of the beneficial microbes are killed.
All right, thank you. And then the next question is, what are the depths that are active in the process?
Can you say that again?
What are the depths, the soil depths that are active in the process?
So basically, what we did, we added the mustard meal, and then we did the rototill. We used the rototill to mix it up. In general, it can vary from crop to crop, but 6 inches should be, because that is the most active, soil layer. We have the root system depth that can reach to up to 6 inches in most cases. So if you have harmful microbes that may have built up from previous crops, if you apply that, and if you want to work with top 6 inches of soil, that should be optimum.
Thank you. So the next question is, are there any non-target effects of the gases produced by the anaerobic microbes, for example, on beneficial microorganisms, soil invertebrates, or other people who are cutting the plastic to release these gases prior to planting?
Very good question. We don't know all the answers at this time, but as has been hypothesized, some of the toxic gases are being produced. The real precaution should be taken by workers is to use similar precaution they are taking when they are cutting the plastic after use of synthetic fumigants. So, in a respirator, it's not, but in general, these gases should not be as toxic as synthetic fumigants, but still precautions should be taken. And what happens due to that anaerobic soil disease infestation to beneficial microbes. We are in the process of enumerating it. We are in the process of making sure that it's kind of difficult to do that, but the science has come in a stage where we can do many things, like metagenomics. We are planning to do some metagenomics to enumerate the microbial DNA in the treated plot and in the non-treated plot. Maybe there is a science that we can also separate the bacterial DNA from fungal DNA, and that the ratio, like we can determine what is in the control plot and what is in the treated plot, especially when we are using probiotic bacterial treatment in our flag plants and other plants.
Okay, thank you. So the next question is, does the amount of water required for the 21 days limit growers in California because of the water cost and availability? Ultimately, this would raise the cost to make application.
That's a good question as well, because these are the things evolving and saturating the soil and then keeping it like that for 21 days obviously will require lots of water when there is a drought kind of situation in California and also in some other places. But I should say that if you are using this and you saturate the soil, this soil moisture will be also be benefiting the plants. And in our case, we actually did saturate the soil at the time when we did the process of anaerobic soil disinfestation. And we used very high impermeable plastic turf, and we did not re-irrigate. So, and the result I will be showing you from some of our other studies really indicates that it still works. So you don't need to irrigate, but still, the situation will determine whether due to water constraint, this process can be used or not.
Okay. All right, great. Somebody asked, what is the average cost of using ASD for the grower?
It really will depend on what kind of carbon source you will be using. Like if you are using grape pomace, like from your local source, that can be very, very inexpensive. On the other hand, if you use rice bran or mustard meal, you have to buy it from out of state sources and then suppliers. In general, I would say if you are using mustard meal for carbon as a carbon source, it can cost you up to a couple of $100 per acre.
Okay.
It's still expensive, but many other companies are trying to make them available to growers.
Okay. Great. And there are other questions, but I know you have more to share. So, so let's move on, and hopefully we can get to those other questions after the rest of the presentation.
Thank you. Yes, so we did very similar work on tomatoes for soil-borne disease control. And as I mentioned earlier, also on eggplants and okra. So I'll briefly show you some of the results. So in the tomato work, we did slightly different treatments. As you can see, these flowers, we had problem with soil-borne diseases, especially we noticed verticillium wilt and fusarium wilt, and also in some cases we had bacterial wilt. So we thought grafted tomatoes on resistant rootstock could be a good option, and we tested that. We also used mustard cover crop, mustard meal. In this case, not typical ASD we used, but we just mixed the mustard meal in the soil. And we used two other biologicals, Freestop and Serinade. Free stuff has a beneficial organism called Glyocladium catenulatum, and Serinate has Bacillus subtilis in it, and obviously non-treated control. So the first data we collected, the plant bigger, after in 20 days, 40 days, 60 days, and 80 days after seeding. So here you can see this Serinate is Serinate black line, and also pre-stop at the seedling production stage, they had higher, bigger. So it's not surprising that they actually enhanced the plant bigger at this time. And for grafting here, you can see after grafting in the blue line here, obviously that due to that grafting shock, plant bigger was slightly lower at this time. But when we did let them grow, and then at the end, you can see actually the grafted plant superseded all treatments in terms of plant bigger. And no wonder that non-treated control had the lowest plant bigger after 80 days when we took the data. So in this case, we used a heirloom variety called Mortgage Lifter. This is a local heirloom variety and very, very productive. Those tomatoes they're like gigantic tomatoes, giant size. And here you can see at the end, we kept track of the harvest from many different harvests. And here you can see the total yield. Grafted tomatoes had the highest total yield at the end of the season. And you can see the 2015 for 2 years of data. 2015 is the blue line and 2016 is the red line. And in 2016, we had to correct some of the issues we got during 2015. And you can see, especially for mustard crop, incorporation was done right. And we saw the significant difference with the non-treated control. But in 1 year, free stock, we did not have the significant difference with the non-treated control. And in 1 year with the mustard cover crop, we did not see any significant difference in yield or significant improvement of yield. But other than that, all the treatments had significantly higher fruit yield in both years, 2015 and 2016. So we also tried to replicate this study with eggplants and okras, as you can see here. For eggplants, we had non-treated check. Once again, very similar. We grew the seedlings in the greenhouse by treating planting mix with beneficial microbes, and non-treated planting mix grown seedlings planted in mustard cover crop field. So basically, in this case, we did not treat the planting mix, only the fruiting field or the field was treated with mustard cover crop. And we tried here pasteurized planting mix, and TerraGrow also applied in the planting holes. So also seedlings grown in regular root shield. Here we used a different product also to compare the result. Here we use the root shield, and seedlings grown on regular TerraGrow treated planting mix, and TerraGrow also applied in the planting holes in this case. And obviously, like previous experiment, we did the combination of treatments, anaerobic soil disinfestation and planting mix inoculation with beneficial microbes. So we also kept track of the plant bigger at the end. You see the 4.8 out of 5, which is the highest plant bigger we obtained from combination of treatment. And we took photos at the end of the experiment. Here you can see number 1 to 6 basically corresponds the treatments. So this is the non-treated, and here you can see the combination treatment. We have seen some diseases in all the treatments, but relatively low disease in combination treatments, ASD and anaerobic and microbial treated plants. So the last trial, actually, we did this, we completed this trial only a few days ago against Verticillium wilt of okra. Okra is a minor crop, but we have seen okra can be very badly affected with this Verticillium wilt. So in this case, from our previous results, we noticed that some of the treatments like mustard cover crop or, like some of the treatments are not that very promising. So we reduced the number of treatment. In this case, of course, we inoculated these plots with Verticillium inoculum. We grew the Verticillium in oat grains and then spread the inoculum in the plots just after preparing the plots. So, and also we grew seedlings in the greenhouse in TerraGrow treatment with regular planting mix. We mixed the TerraGrow in the regular planting mix, and also we pasteurized. So just wanted to make sure that we are getting any extra benefit by pasteurizing the medium, but from our previous results, we did not see any significant difference in pasteurization and non-pasteurization of the medium, which is a good news for the growers because, in our experimental setting or in the university, we can do the pasteurization very easily of this growing planting mix, but in growers' condition, it may not be possible for them to do it. So we found like just inoculating the regular planting mix with beneficial microbes may be enough. And in this case, we did the combination of this planting mix inoculation and anaerobic soil disinfestation. So we had 3 different treatments: non-treated control, planting mix, regular planting mix inoculation, and ASD, pasteurized medium, and ASD. So here you can see the photos we take at the end of the group. So let me go back and show you how the plants actually started wilting in the non-treated plot. We lost more than 50% of the plants in the non-treated one. And in the treated plots, only 1 plant in 1 treatment. So this is very significant. And also at the end of the season, when we went back and took the photo, you can see the thinning of okra canopy in the non-treated plot. And in that treated plot, here you can see the anaerobic soil disinfestation and planting mix inoculation. The canopy was significantly retained, the canopy very thick even at the end of the season, and very similar with the other treatment as well. We also collected yield data from these treatments. As here you can see the number of fruit per plant, 52 compared to 67 when we used pasteurized, and the regular mix, 72. So significantly higher in the treated plots per plant fruit. So this is a good news because we just wanted to make sure that pasteurization of planting mix make a difference, but here we are seeing that actually it did not provide any benefit, rather it affected the performance. So what happens when we do the pasteurization of medium? What we notice that it gets really, the medium gets really dry and hydrophobic. So, and I put this, in this case, I wanted to mention that we also treated the seeds with this TerraGrow. So when we put the seeds in the medium, it does initially the seedling, germination can be affected in pasteurized medium because the mix can be very, very dry. And the fruit weight here you can see also significantly higher by weight, compared to 45 ounces compared to 60, and 62 ounces per plant. And the average plant height was not significantly different in these two treatments, but regular mix had significantly higher plant height as well. But the most striking here, you can see that, like I mentioned here, non-treated, we lost more than 50% of the plants at the end of the season. In pasteurized, only one, and in regular mix, we did not lose a single plant, which is very, very significant. And I am very optimistic to say that if you can adopt this technology, growing your seedlings in beneficial microbe inoculated planting mix and take it to the field where you can do the anaerobic soil disinfestation and do the planting in those treated plots, you will see a difference in, especially when you have a problem with soil-borne diseases or soil-borne organisms in your soil. So with that, we are very close to an hour mark.
Yes, we are. So we have some great questions. So what I'm going to suggest that we do is that we answer those questions and go over to 12:15, probably by the time we've wrapped up with everything. And for folks who need to jump off, there is a recording, and that there's some good questions. And so I think it's worthwhile answering those. So I'm just going to kind of go through them here. Could the ASD technique be used for anaerobic composting with appropriate arrangements? For example, cubes with scrap materials covered with tarp?
I would say it's probably not needed.
Okay.
Because when growers, especially small growers and gardeners, they have disease issues, we always recommend that they should not take those diseased plant debris to their compost pile. Otherwise, when you are doing the composting process, this is some of those microbes. Not necessarily those are all harmful microbes. In those cases, there are many beneficial microbes. They can multiply in compost. And then when you are taking that compost in your field, you are not only adding biomass, also you are adding in most likely some beneficial microbes in there. So I would not suggest doing anaerobic disinfestation or anything like that. But during the composting process, when you are doing it right, the temperature can go very, very high. I don't remember the exact temperature, but it's probably 135 degrees Fahrenheit or so. And that can actually kill many of the microbes. So in some cases, some growers, they used diseased plant debris, and then if they do it right, those microbes, harmful microbes, can be killed anyway. So I have not heard of anybody suggesting or doing anaerobic soil disinfestation during the composting process.
Thank you. Have you published these results anywhere yet?
That's a good question. And what we are doing actually, by seeing that very good result from the okra trial, we did a video clip just a few days ago, and it will be followed with a fact sheet. We will be publishing that very, very soon in our extension website. And strawberry data, we are submitting it as a peer-reviewed manuscript in plant disease. So hopefully that one will be published also in a couple of months.
Great. So does ASD work differently in different soil types? For example, the soil in Florida is very sandy, while a lot of soil in the Northeast has quite a bit of clay. My understanding is that even synthetic fumigants may be more difficult to use effectively in heavier soils.
Very good question, and that's probably the reason we have been experimenting these in many different geographic regions. That's absolutely right. Like when you're talking about nematodes, in our heavy soil, nematodes problem is less than sandy soil in Florida. And how anaerobic soil is infestation will, efficacy will vary in different soil types. I think it will be different, but as we get more results from different agroecological regions, we may compare those results and come to a conclusion, what it is effective more in our Northeast heavy soil, or it is more effective in the Southeast. And that's I think the whole objective for Northeast SARE and the Northeast IPM Center for giving so many grants on these kind of new technologies. I know many grants have been approved for trying in many different soil types. So at this point, we don't know for sure, but I think there will be a difference.
Great. All right, we have 4 more questions. So does prolonged use of ASD become ineffective due to the selection of resistant pathogens?
No, the answer is no, because say resistance issue is a big issue for plant disease management, and that is an issue for many different types of fungicides. Mutation in plant disease organisms can happen, but in this case, as I said earlier, we don't really know the exact mechanism how it works. So think about fumigation, synthetic fumigants, when growers used to use methyl bromide, that would indiscriminately kill all kinds of microbes, insects, and weed seeds. I believe if the same methodology works here, resistant biotypes will not be an issue here.
Okay. All right. So there's a question: what methods are being considered for enumeration of microbiome in the rhizosphere?
That's a good question. And, we used, in fact, from our trial, we collected the soil samples from the rhizosphere, and we basically did the soil dilution plating to enumerate fungal colonies and bacterial colonies. So this is kind of crude way to say, okay, in this treatment, we had more bacterial colonies than fungal colonies. And we can say more of these, if fungal colonies, some of these are harmful microbes. And if they are more, we don't know these are, because we don't know these are harmful fungal pathogen or fungal colonies or beneficial fungal colonies. So that has not been separated. But there are molecular techniques, like I mentioned, metagenomics, like that can extract the whole DNA, and then we can amplify bacterial, 16S, the ITS region and fungal ITS region to separate and then say, this is mass from fungal and this is from bacterial. But still, we will not probably get the answer, like whether it's beneficial or harmful. But as we used all Bacillus type of probiotic bacteria in our trial, and we have the soil sample to analyze it, if we get something to specifically identify that these are Bacillus bacteria, different species of Bacillus, then we can answer some of those questions, but it will take time.
All right, thank you. We have 2 last questions. So the first is, can beneficial microbes colonize the fruit and cause harms to human in strawberry fruits?
Answer is no.
Okay, great.
There are many different types of products with these beneficial microbes, and people have been using those, and these are not harmful to, and all these fruits are edible.
Great. And then the last question is, did you apply fungicides during your study on several hosts?
No, we did not use any fungicide. So basically, if you would see, so we talked about two different types of diseases, right? For foliar disease and soil-borne disease. In this case, we used we did these trials for soil-borne disease control. If you would see any big issue with foliar, pathogens that would impair our study, that we would use fungicides for controlling foliar pathogens. But in all these cases, actually for the strawberry trial, we conducted in a certified organic farm. So there is no question of using any synthetic products there.
Okay.
We did not use any.
Wonderful. Well, thank you for your wonderful questions, and thank you, Mahfouz, for your wonderful responses. And a couple of people have said thank you in the Q&A. So we do have another quick poll for you to assess change. So if that poll can pop up, and before you jump off the call, if you could answer those questions, that would be wonderful for us. So thank you. And I'll be quiet while that happens. And then Mahfouz, if you could just roll the slide forward for me, that would be wonderful. There we go. All right, I will share the results. And I won't go over them one by one, but you can see that people's knowledge has shifted from not at all into the somewhat to moderately knowledgeable range. And the most interesting one, I think, is as a result of this webinar, how likely are you to use biofumigation ASD to manage soil-borne diseases? And we've got quite a lot of somewhat moderately likely and very likelies. And increase your use of IPM, which is what we love to hear. So thank you very, very much. So before we conclude, I just want to share a couple of things. Thanks to the funders of this research work. And next slide, please. Thank you. We have a couple of upcoming webinars and more are being added, so please keep your eyes out. We've got one on cooperative extension in Indigenous communities in November, and one on non-traditional areas for IPM careers with 2 LGBTQIA plus individuals in December, and more are being added each day. So please feel free to join us for those. And we've already gone through that. If you would like to participate in some of these research or connect with colleagues, we do have a mechanism for that on our site. You can put up a profile of who you are and what you're interested in, and then you can also find people in the Northeast who are doing work and connect with them. So actually there is not a slide for this, but our RFAs are out at the moment. The deadline is November 15th. And if you are interested in applying for one of our grants, please go to our website and check out the 2 RFAs that we have available. So next slide, please. The recording will be available next week. I'll send that to you. And the next slide, please. And we want to acknowledge that the Northeastern IPM Center is based at Cornell University in Ithaca, New York. Cornell University is located on the traditional homelands of the Gayokono, the Cayuga Nation. The Gayokono are members of the Haudenosaunee Confederacy, an alliance of 6 sovereign nations with a historic and contemporary presence on this land. The Confederacy precedes the establishment of Cornell University, New York State, and the United States of America. We acknowledge the painful history of the Gayokono dispossession and honor the ongoing connection of the Gayokono people, past and present, to these lands and waters. This land acknowledgement has been reviewed and approved by the traditional Gayokono leadership. And the next slide, please. And we also want to acknowledge USDA NIFR, who provides funding for the center and for our research grants and for projects like the one you've just heard about. So it is without their funding, this wouldn't be possible. So with that, I will conclude the webinar. Thank you for your participation. Thank you for your great questions. And thank you, Dr. Rahman, for all of the work, hours of study and concentration and learning that has gone into to this research and your ability to share it so beautifully with us today. So thank you.
Thanks, Jenna, for the invitation, and thanks to the Northeast IPM Center.
Okay, terrific. And I see that there's a couple of other questions that have come in. Actually, people just saying thank you. And the person who asked if the PDF of this presentation is available, yes, it will be. When we put up the recording, we also put up a link to the slides. So you'll see that it's usually just above the video recording. All right, wonderful. Enjoy the rest of your day, everybody. Okay, bye-bye.